Hello everyone, my name is Nandini Shukla and I am a student from Biotechnology of first year. In this video, I am going to talk about a microbiology topic which is horizontal gene transfer mechanism. So, what is horizontal gene transfer? It is a movement of genetic material between unrelated organisms not through the normal reproduction. So, in this video, we are going to see its introduction, its history, mechanism, method of detection, significance, conclusion, and the reference. Let's begin with the introduction part. Gene transfer refers to the movement of gene within or between the individual organism. HGT is often used to refer to all forms of gene transfer that do not involve parento-offspring transfer, that is sexual or asexual. It can occur either naturally or by human intervention. Example, gene technology, which involves manipulating an organism's DNA to study, alter, or transfer gene. Embryo rescue is to save and grow immature or weak embryos. In vitro fertilization, that is IVF, is a fertility treatment where an egg is fertilized by a sperm in a lab rather than in a human body. Protoplast fusion is a plant biotechnology to form hybrid cell, and self-cloning refers to natural or sexual reproduction in organism or artificial method which creates genetic copies. Each form of HGT comes with different consideration of risk, which is the case of genetic modification or genetic engineering, commonly regulated through legislation. So this was the introduction of HGT. Now we are moving about his history. Which scientist discovered it? The scientist Frederick Griffith. He reported his experiment in 1928, suggesting first experiment that bacteria are capable of transferring genetic information through a process known as transformation. Griffith's findings are followed by a research in the late 1930s and early 1940s that isolate DNA as the material that communicates this genetic information. This is a chronombacterium diphtheria. Horizontal gene transfer was then described in Cetale in 1951 in a paper demonstrating that the transfer of viral gene into chronombacterium diphtheria create a virulent strain from a non-virulent strain, simultaneously revealing the mechanism of diphtheria and giving the first example for the relevance of lysogenic cycle, that patient could be infected with the bacteria but not have any symptoms and then suddenly convert later or never. Interbacterial gene transfer was first described in Japan in 1959, publishing that demonstrate the transfer of antibiotic resistance between different species of bacteria. In the mid-1987, I postulate that biological significance lateral gene transfer has existed since the beginning of life on Earth and has been involved in shaping of all evolutionary history. So, this was an overview of history of horizontal gene transfer mechanism. Now, moving to the mechanism of horizontal gene transfer, there are three different mechanisms: transformation, conjugation, and transduction. First, I will explain in a very short what is this three type, then we will go in detail about these three types. So, first, in transformation, a bacterium absorbs naked DNA fragment from its environment, often from dead cell. This makes the recipient cell capable of expressing new gene. Next, conjugation, a transfer of genetic material between two living bacterial cell via physical bridge happens. That is, here we can see that a physical bridge which is known as pilus. A donor cell, which is F negative, extends to pilus and recipient cell, that is F positive, and transfer a copy of its plasmid, making the recipient a donor too. Next, in transduction, a virus accidentally packages DNA from one bacterium into new particle. When this phage infects another bacterium, it injects the previous host DNA, transferring the genes. So this was the short overview of this mechanism. Now we will move in detail about this three types. Let's begin with the first mechanism, that is transformation. So I'm going to explain you in short and in easy manner by using this picture of transformation process. So let's begin. So first, a bacterial cell dies and bursts open. This process is known as cell lysis. When the cell breaks, it releases DNA in the environment, and these DNA fragments are released into the environment. You can see the cell lysis, the DNA fragments which are scattered in the environment. Next, a nearby component bacterium comes into contact with this free DNA. Component bacteria have special proteins in their cell membrane and uptake machinery, which help them recognize and take in the DNA from the environment. The DNA then passes through the cell membrane and enters the bacterial cell inside the cell. This upcoming DNA combines with bacterium's own chromosomes or plasmid through a process called homologous recombination. As a result, the bacterium gains new genetic information. This new DNA may provide useful traits such as antibiotic resistance or improved survival ability. So, in short, we can say that a dead cell releases its DNA, a component cell takes it, and the DNA integrates into genome, and a new traits appear in this. So, this was the process of transformation. Now, we are moving to the next mechanism, that is conjugation. So, in this process, the donor cell contains a special DNA molecule called conjugative plasmid. So, here you can see this is a conjugative plasmid. This plasmid carries the gene required for DNA transfer. The recipient cell initially does not have the plasmid. So, you can see here the recipient cell does not have its plasmid. The donor cell forms a thin tube-like structure called pilus. These pilots connect the donor cell directly to the recipient cell, allowing contact between them. Though the pilus, a single-strand plasmid DNA is transferred from the donor cell to the recipient cell inside both cells, the transferred DNA strand is replicated to form double-strand plasmid cell. As a result, after conjugation, both cell contains plasmid. The recipient cell now gains new genetic traits and can also act as donor in future conjugative events. So, in short, we can say that conjugation is a direct transfer of plasmid DNA from a donor to a recipient bacterium through a pilus. Next, we move to the third and the last mechanism, that is transduction. So, in this a bacteriophage, we can see here a bacteriophage infect a bacterial cell and inject its DNA. Inside the cell, the phage replicates. During this process, a mistake can occur in this fragment of the bacterial chromosomal DNA is accidentally packaged into new phage particle instead of phage DNA. Next, the infected bacterial cell undergo cell lysis. Here you can see this is going a cell lysis, releasing many new phage into the environment. Some of this phage now carry bacterial DNA. This phage then infect another bacterial cell and inject the bacterial DNA that they are carrying. Inside the new host cell, this DNA integrates with the bacterial chromosome through homologous recombination. As a result, the recipient bacterium gains new genes even though there was no direct contact between the two bacteria. The key point is that DNA transfer occurs via virus, and this process is called generalized transduction. So, in short, we can say that transduction is the transfer of bacterial DNA from one bacteria to another by bacteriophage. So, this was the three mechanism of horizontal gene transfer, which was transformation, conjugation, and the last transduction. So, by completing this mechanism, we are moving to its method of detection, how it is detected, how it's happened. Horizontal gene transfer is typically inferred using bioinformatics method, either by identifying a typical sequence signature, that is parametric methods, or by identifying strong discrepancy between the evolutionary history of particular sequence compared to that to their host. The transferred gene found in the receiving species is more closely related to the gene of the donor species than would be expected. So here you can see a picture. I'm going to explain this picture you in detail and you will understand what exactly is happening in method of deduction. At the beginning you can see a simple ancestral gene called gene A. When evolution occurred through speciation, the ancestral gene splits into two different species, that is species 1 and species 2. This process represents vertical gene transfer where genes are passed normally from parent to offspring. After speciation, species 2 carries only one copy of gene, that is gene A2. However, species 1 contains two related genes, that is gene A1 and gene A2. This difference is due to normal inheritance. Instead, species 1 acquired an extra gene through horizontal gene transfer. In this process, genetic material is transferred between organism and not parent and offspring. This commonly occurs in bacteria through transformation, transduction, and conjugation, the three mechanisms we have seen earlier. This diagram clearly shows that because of horizontal gene transfer, gene evolution does not always follow species evolution. As a result, gene trees may differ from species tree. In conclusion of method of deduction, I can say that horizontal gene transfer plays an important role in genetic diversity and evolution, especially in microorganisms. So this was the overall process of method of deduction and the cycle where gene A is converted through species 1 and species 2, and in species 1 there are two genes because of horizontal gene transfer, and species 2 have only one gene, that is gene A2. Now we are moving to its significance. So there are different types of significance. Evolutionary. First, the evolutionary significance. In this we have three types: accelerated evolution, adaptation and innovation, and next is shift the trees of life. Let's begin with the evolutionary significance. So, accelerated evolution. HGT provides a faster way to acquire beneficial gene like new metabolic pathway or stress tolerance than traditional mutation and vertical inheritance. Adaptation and innovation. It enables rapid adaptation to new environments, creating biological novelties and driving biodiversity in prokaryotes and eukaryotes. Shift the trees of life. Instead of simple tree, HGTCAM makes the evolutionary history of microbes look more like a web, with gene moving freely between distant lineages. Next, we have medical and health significance. In this, we have antibiotic resistance, pathogen virulence, and disease development. In antibiotic resistance, HGT is a major reason bacteria quickly develop resistance to new drug, making infectious harder to treat. Pathogen virulence. It allows pathogen to acquire virulence factor, increasing disease severity. Disease development. HGT can contribute to genetic genetic disorders, cancer, and neurodegenerative disease, but not understanding it helps develop diagnoses and therapies. In significance, next we have agriculture and biotechnology significance. Crop improvement. HGT can transfer beneficial traits like disease resistance, example fungus resistance in wheat, from wild relative or microbes to crop. Next we have genetic engineering, that is HGT mechanism like conjugation. The three mechanisms we have seen are used as tool to introduce desired gene in biotechnology and gene therapy. Next is ecological significance. In this we have microbiome dynamics, that is HGT influence interaction within microbial communities affecting ecosystem function through gene sharing or cooperative or competition. Next we have symbiotic relationship, which is it facilitates the exchange of gene that shape beneficially or harmful interaction. So this was the significance of horizontal gene transfer. Here we have pivot mechanism for horizontal gene transfer. In this picture, you can see first a cuscuta with its host, second a hornwood and a firm gametophyte, and in the third picture there is a C4 grasses. So what exactly is happening in this picture I am going to explain. So cuscuta and its host. So cuscuta is a parasitic plant. It forms a special structure called hostoirism that penetrates the host plant tissue. Through this innovative physical connection, DNA or RNA can move from host to cuscuta or vice versa. This close cell-to-cell contact provides a direct pathway for gene transfer. That is, parasitum enables HGT due to shared vascular connection. Here, the white arrows show how the gene transfer is taking place. Next, in the second picture, a hornwood and the firm gametophyte shows the interaction between gametophyte stage of plant. Gametophyte are thin, simple tissue with less structural protection. Close contact between gametophyte may allow uptake of foreign DNA. The insect highlight transfer at the cellular level. So, in this, HGT may occur during earlier exposed life stage of plant. Next, in the third picture, we have C4 grasses. Here, it shows grass reproductive structure, that is flower. HGT is proposed occurs via pollen-mediated transfer or hybridization-like events. Gene related to C4 synthesis may spread between grass species, that is, reproductive tissue may occur as a route for gene transfer. So the overall significance is HGT is not limited to bacteria, and plants can acquire gene through parasitism, physical contact, reproductive interaction. HGT also contributes to adaptation, evolution of new traits, and metabolic innovation. Example, C for photosynthesis. So this was the overall significance of this. Now coming to the conclusion of horizontal gene transfer, we will conclude this by several points, as you can see here. So it is important to know about the difference between each of the three types of horizontal gene transfer. They all share a few features in common. Second, bacterial cells only keep double-strand circular DNA. Any single-strand linear DNA that are acquired from horizontal gene transfer must be incorporated into plasmid or chromosomes and replicate, so it is a double strand. Any DNA that is left linear or single strand will be degraded. Bacterial cells are prokaryotic. They are normally haploid and contain only one copy of each gene. Bacterial cell also only contain one circular chromosomes but can have multiple smaller circular pieces of DNA called plasmids. When a bacterial cell receives a copy of gene it already has, it may integrate the new copy into its chromosome or a plasmid. Horizontal gene transfer can make a bacterium a partial diploid, a cell that has one copy of most gene but has two copies of few genes. These two copies can be identical but could also be different alleles for the same gene. Typically, the new allele will be stored in a plasmid. Both copies of gene may be expressed and then alter survivability. So this was the conclusion of horizontal gene transfer mechanism. So now I am going to share the reference from which I have taken this all material. The different websites are Wikipedia, University of Colorado Denver, which is in the form of PDF, Technological Networks, PubMed Central. So these are the different references from which I have taken this material and made this presentation. So this was all about the horizontal gene transfer mechanism. Thank you.